Physiology of muscles. Muscles may be divided into skeletal striated muscles, skeletal muscles, smooth muscles, and the cardiac muscle. Cardiac muscle we will discuss in the special lecture. Here we focus on the skeletal muscle and the smooth muscle. So let's speak about the difference between skeletal muscle and the smooth muscle. Skeletal muscles are responsible for the physical activity, for walking, for movement of limbs. Smooth muscles are found in the internal organs, and they are producing the function of intestine, of the blood vessels, and so on. The structural unit of the skeletal muscle is the muscle fiber. Smooth muscles consist of cells, which are called myocytes. Skeletal muscles are controlled by the somatic division of the nervous system. Smooth muscles are controlled by the autonomic nervous system. Smooth muscles are characterized by the automaticity. Automaticity is the ability to excitation without any external stimulation. So the source of the stimulation is inside the cells. Skeletal muscles are rapid. One single contraction duration is about 0.11 seconds. Smooth muscles are moving slowly. They are slow. One contraction may be 20-30 seconds or even more than 20 seconds. Smooth muscles can be contracted for a long time without fatigue. Low fatigability. Skeletal muscles get fatigued soon after the beginning of the work. High fatigability. And the smooth muscles are highly sensitive to the chemicals, to drugs, to hormones. If we will apply the drop of acetylcholine on the smooth muscles, we will observe the reaction, and we will not if we will apply acetylcholine on the surface of the skeletal muscle. Now we will speak mostly about the skeletal muscles, but we will take into consideration the peculiarity of the smooth muscles. There are, for example, the source of calcium in the skeletal muscle is endoplasmic reticulum. In the smooth muscles, calcium is extracellular. There are peculiarities in the proteins which are responsible for the muscle contraction. In the smooth muscle, we have calmodulin, calcium binding protein. In the skeletal muscle, it's troponin. But now we will speak about the mechanism of the muscle contraction. On example, for example, we will take the contraction of the skeletal muscle. So we will draw the graph of a single muscle contraction first. So muscle contraction begins after the stimulation. We indicate the stimulation by an arrow. And the first phase of a muscle contraction is called latent phase. Latent phase. At that period, only excitation of the membrane of the muscle fiber may be measured. No contraction. So first phase is called the latent phase. So we have no contraction. Second phase begins after this excitation and is initiated by excitation, is called shortening period or shortening phase. Muscle shortens, so this graph indicates the difference of in size of a muscle. So first phase is shortening phase, and the third phase is called relaxation period, relaxation phase. So we have three phases of a muscle contraction. First phase is about 0.01 second, 10 millisecond. Second phase 0.05, 50 milliseconds. And the third phase 0.05 second. So totally summarizing, we'll have the duration, average duration 0.11 seconds for the striated muscle. Next question is the electromechanical coupling. Electromechanical coupling, that is the mechanism that is converting the electrical process on the membrane, the depolarization of a membrane, into the mechanical processes, shortening and relaxation. So now we will discuss the electromechanical coupling in the muscle contraction. The muscle fiber, this is the surface of a muscle fiber, is stimulated by the nerves which produce synaptic contacts. We have vesicles with acetylcholine that is produced, released, binds the receptor on the surface of a muscle fiber and induces the sodium influx and the depolarization of the muscle fiber. This depolarization activates the sodium, activates calcium channels, which may be found on the sarcoplasmic reticulum, that is the depot, that is a source of a calcium. So calcium is the most important player that initiates the electromechanical coupling. So calcium is released from sarcoplasmic reticulum. This reticulum may be called L tubules. L tubules, longitudinalis, and transverse T tubules, which are in fact the invagination of a membrane into the fiber, so that action potential may easily go into the muscle fiber and initiates the calcium channels. So, therefore, the calcium concentration rises 1,000 times in comparison with the resting state. So, calcium is responsible for the electromechanical coupling. So, what calcium does actually? To discuss this topic, we should say that there are specific proteins in the muscle. We have two proteins in the skeletal muscle that directly involved into the contraction. It is the protein myosin, and protein actin. Myosin and actin, they produce the filaments, thin and thick filaments. So let's illustrate it. So we have again the muscle fiber that is divided by Z lines onto the sarcomeres. Each sarcomere is separated by two Z line, and that is also the Z line. The thin filaments, actin filaments, are attached to the Z line. So thin filaments are attached to the Z line. Here, 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 and so on. And between the actin filaments, we have thick filaments, or the myosin filaments. This is the thick, and again thick. So this is myosin. Filaments. The myosin produces the so-called heads. This is the myosin heads that are crucial for the sliding of actin and myosin filaments past each other. So the filaments may pass each other when the angle of the myosin head changes. This change of the myosin head is called oar stroke. So like when the boat is moving in the river, there is oar stroke. So in the same manner, the myosin head may change its angle. So then, what is the role of calcium? So calcium binds to the protein troponin that lies between the myosin head and the actin. So when calcium binds the protein troponin, then another protein, tropomyosin, opens the access to myosin head to protein actin. So when calcium is absent, myosin head is not able to attach to the protein actin. because troponin and tropomyosin, they block the excess. But when calcium is on the scene, is present, then troponin and tropomyosin, they change their conformation so that at that point the myosin head binds actin, and actin is inducing the break of ATP. So on the myosin head there is ATP, and this ATP is broken by the ATPase, and the angle of the myosin head changes, and at that moment the filaments slides. So we call this concept sliding filaments theory, that explains the shortening of the sarcomere and of the muscle fiber as a whole. So electromechanical coupling is initiated by the excitation of the membrane, and then by the release of calcium that initiates the molecular processes with the troponin, tropomyosin, the proteins that control the muscle contraction, and with actin and myosin, the proteins that produce the sliding filaments, and the muscle shortens. We discussed already the single muscle contraction, but another type of the contraction is called the continuous muscle contraction, or tetanus. So let's speak about the tetanus. In that case, we have not one stimulation, but a number of the stimulations with high frequency. So let's indicate the first stimulation, and we have the single muscle contraction. So we have latent phase, shortening, and the relaxation phase. So if another stimulus hits when the relaxation phase is over, we will have another single muscle contraction. But if we will increase the frequency, and another stimulation will hit when we have the relaxation phase of the previous contraction, then the muscle will contract, not completely relaxed. So it is called the staircase tetanus. Staircase. If we will increase the frequency of stimulation in such a manner that the new stimulus will hit when muscle is not even begins to contract, but at the shortening phase we will have the tetanus phase. At that point the muscle contracts even more, amplitude increases even more, and at some frequency we will have the maximal amplitude of the muscle contraction. If we will increase the frequency even more, we will have complete relaxation. This phase is called pessimum, and the highest amplitude of the muscle contraction is called optimum phase. Let's explain optimum and pessimum. So, when the new stimulus hits at the exaltation phase, so let's indicate it on the first contraction here. So exaltation phase, it is quite high frequency, then all muscle fibers are very excitable, and we will have optimum. If we will increase the frequency even more, the new stimulus will hit at the refractory period. refractory period. At that point, the muscle will not be able to contract and will completely relax. So, repeat it again. If the new stimulus hits at the relaxation phase of the previous contraction, hits here, then we will have staircase tetanus. If the muscle is stimulated by the second stimulus at the contraction phase, we will have tetanus phase here. If the muscle is again stimulated at the exaltation phase, we will have optimum. And if the muscle is stimulated at the refractory period, here, then we will have pessimum, muscle will completely relax and will be inhibited. Now let's discuss electromyography. So electromyography is the method of investigation of muscles. It is a method of registration of the electrical activity of the muscle. So electromyogram looks like that. If the muscle performs a work, then amplitude of electromyogram increases like that. So here is rest, here is work. Electrical activity of a muscle is the sum of action potentials. Each action potential is produced by a single muscle fiber. So here we have the summary of all action potentials in that muscles. That's why when the muscle performs some work, the new muscle fibers are involved in the activity and the amplitude of electromyogram increases. Here we should say that the skeletal muscle doesn't obey the law all or none. So if we will increase the amplitude of the stimulation, then we will see that subthreshold stimulus will not induce contraction, threshold small contraction, and then the amplitude will increase till it will reach some maximal amplitude. So this is the gradual response, not all or none. Each muscle fiber obeys the law all or none. But each muscle fiber has its own threshold. That's why when the work increases, new muscle fiber, less excitable, are involved in the contraction. Smooth muscles partially obey that law all or none. Cardiac muscle obeys law all or none and contracts as a whole unit each time. Muscle fibers may be divided into red and white fibers. Red fibers they contain myoglobin and are able to contract for a long time without fatigue, but are not able to skilled movements in comparison to white muscle fibers, which get fatigued easily and rapidly, but are able to fast and skilled movements, but not, as it was told, able to work for a long time. Muscle contractions are divided into isotonic, divided into isotonic and isometric. During isometric contraction, the length of a muscle stays constant. The tonus, the strength, the tension changes. The law of medium load. The medium load law. This law states that the maximal work is achieved when the load on the muscle is average. So when the load is too high or is too small, too low, then the ability to produce work decreases. So at medium load, the work is maximal. How it can be explained? So let's imagine that we have 3 loads. The low load, the medium load, and the high load. So here we attach to the muscle, 100 kilograms, here 5 kilograms, and here 5 grams. So let's see at the sarcomere. And the medium load, the position of thin and the thick filaments is optimal for myosin heads to attach the actin and produce cross bridges. If the load is too high, then the muscle elongates and the actin and myosin filaments cannot produce the sufficient number of cross bridges, so the work decreases. And if the load is too low, then actin filaments overlaps, and again the sufficient cross bridges are not produced. So therefore, at medium load, the muscle contracts with maximal work ability.